My name's Cathal Garvey. I am an independent researcher or biohacker, depending on the audience, but I'm not the first person to break the term, so I'm just going to use it with good comfort. I'm a biohacker. And I don't just use that word to be provocative. It's actually sort of an identity or a lack of identity thing that, of course, Bill was alluding to the early computer science breakout into the entrepreneurial sphere and also just into the sort of experimental sphere outside of an academic context. And that's where the term hacker came from. It wasn't originally a term of a pejorative term. Yeah, it was originally a term of respect. That a person who can do a lot with a little or do something remarkable or unexpected might be called a hacker by his peers. And it's still used that way by people in that community. And, you know, we kind of decided as a community, a lot of people in Europe decided to just start using the word biohacker and damn the consequences and say that people are going to get around to it and stop worrying about the word hacker. Now, before I go into this, because I might forget to say it, two things. Firstly, you may get the false impression that I am a loner and that I want to work alone. People have gotten this impression, and it's quite false. I would be very welcoming of other biohackers to work with in Cork. It's merely been the lack of, in my small circle, my little bubble, I hadn't heard that there was an iGEM team until very recently. I hadn't heard that there was going to be this industrial park opening with synthetic biology in its list of keywords. So actually, the other thing then is, if anyone in Cork or the broader Ireland wants to set up a biohacker space, I'm eager to help, and I've already gone through the regulatory hurdles and gotten my own license, so I can help. So this is my lab, which is licensed. Well, it's a small corner of it. This is a spare bedroom in a house out on the Lee Road. It's not my house because I live in a much smaller house in Ballyfihan. It is my very tolerant parents' house. They have a biolab in their house that they have to deal with. And I've been able to work in this lab for the last few years, initially on my own savings, and then after a fortuitous meeting with the assistance of SOS Ventures and Bill Lau, I've been able to try and focus on my sort of personal mission, which is, you know, with mixed results, to try and make open source biotechnology a thing. And why is, since we're talking about translation, which is, you know, bringing ideas from concept to market, why do I think open source needs a boost right now? Because in the early computer science market, I use the word market, in the early hacker movement, I suppose, there wasn't nearly as much of a patent and intellectual property thicket as there is today, in both fields, in fact. If the hacker community were trying to start home computers today, they would have greatly more difficulty due to the growth in patent trolling and the thickets. I mean, the system has degenerated. But thankfully, before that happened, we got in open source. And open source made all the difference. Now you can quickly, easily set up a fairly secure product in computers because of all the work done by other people before you. And most of the people in this room probably have an Android device. Swept the world, has changed the way that people use phones, and that is based on open source stuff. And I feel there's not enough of that in biology. I think it's a critical thing that we need to address, and I decided, okay, while I'm at it, why shouldn't this be fundamental stuff that makes it easy for anyone to pick up synthetic biology and do it at home? If we're always talking about bringing synthetic biology into the day-to-day lives of people or into their livelihoods, they need to have tools that are designed for that. It's not enough to design something using and requiring the methodologies of an academic lab and then say, job done. Just do what I did. Nobody can do what you did. You're working in one of the most exceptionally well-funded labs in the world, relatively speaking. Any first world lab is a princedom of biotechnology. So if you expect people outside that lab to be able to do what you did, it won't happen. So this is my lab. I kind of chose this picture to show that I actually do work in this lab. It's not the tidiest picture. And a quick glance around the picture, if you chose to, you'd see like lots of cobbled together stuff. In a way, that's philosophically deliberate for me. I feel that, again, if the hardware can't be bought, borrowed, knocked together, or built using whatever methods are available to you, you should write it off. Now, long before this was sort of a profit-making enterprise for me, I was still kind of working in an academic lab on a more conventional job and got into the hobby of 3D printing and also started encountering this DIY bio thing. And I said, one of the pieces of hardware that no one's addressed yet is a centrifuge. Most people in this room don't need an introduction to that. So I knocked something together that I could print on my own 3D printer which you could attach to a multi-tool called a Dremel, called it Dremelfuge, and uploaded it onto the internet. And I was sort of blown away by the enthusiasm that people had for this, that they were like, this is amazing. People still think that this is a kind of a real nice thing. It's two silly technologies, 3D printing and synthetic biology. So, well, it's a tool in synthetic biology. So you can print this out, and this is also a gel rig that I had designed that you can print. So someone printed this elsewhere in the world and sent me a picture. And you can stick that to a tool, and you can put your samples into the rotor and fire up the tool, and now you've got a centrifuge. And there's a lot you can't do without a centrifuge. So, this was, as simple as it is a really foundational thing that is now no longer hundreds of euro. You've probably already got a Dremel or a tool. Print this out on your friend's thing or order it from the internet, and now you've got a centrifuge, no problems. And I wasn't the only person. And actually, this is why I feel that, why are you all so interested in this terrible centrifuge? I mean, there are people doing way more amazing things. This is OpenPCR. It takes thermal cyclers, which would never have been gotten for less than several thousand, and it made one for several hundred euro. And it's an open source hardware piece of equipment that you can use to do DNA fingerprinting, to do DNA assembly, to do the workhorse reactions in synthetic biology. I've got one of these, and it works fine, fantastic. It's just a PCR machine. People have gone all the way up to doing DIY bioprinting using repurposed is it inkjet or laser cartridge? Inkjet cartridges. So this is an inkjet cartridge, some open source hardware, Arduino, open source shields for the Arduino, and you just cobble it together. And the guys in BioCurious, a biohackerspace in San Francisco, well, I should say the Bay Area, it's not some complicated geography over there, they made themselves a bioprinter. So this is 3D printing, it's so old, no, we're printing cells now. So when I had a bit of stuff put together, and I had found that the academic work that I was doing had drifted away from my area of interest, and I also felt this is too important to let slide. I want to do this full time. I want to give this a chance. So I said, all right, I'm going to try and make the first open source cloning plasmid built by and for people outside of an academic context. And I made all my first mistakes on this baby. This is my learning experience. So I said, okay, not E. coli. E. coli was always more appropriate for biotechnology, for well-funded labs. Let's do this in Bacillus subtilis, which is way easier to grow, maintain, store, all this. I still believe Bacillus subtilis is great, but I'm no longer developing for it because it's an additional level of difficulty. And actually, this was death by 1,000 cuts. All the different things I wanted to translate into the market. As Bill will advise now, do the minimal thing. Do the thing that will actually work. And I was like, no, this has to be antibiotic-free. It must not need antibiotics. It must work in Bacillus subtilis because it's an easier thing to use. It must be BioBrick compatible. So I took a backbone, I engineered out all of the different restriction enzyme sites so that I could put them into a multiple cloning site, which involved hand design of different crazy secondary structures. I dealt with the stability issue of most cloning plasmids where they get lost in the absence of selection. No, throw back in that thing that people accidentally chopped out in the 90s when they didn't know what they were doing. And then I put in something which actually, to my pride, did work. I said, okay, let's put something in there that when there's sugar present, the cells run away. They become motile, which Bacillus lab strains normally aren't, and they run away from it. And I called it Sugar Rush. And you take the cells, you add some sugar solution, you dot them in the middle of your agar plate, and you go away for the night and come back tomorrow. And you'll have all these little satellite colonies that have gone and run away. And when the sugar is far behind them, it turns off and they stop and form colonies. And I'm proud to say that actually worked. Which, a friend of mine called this a moonshot, and I was like, I'll show you, and I showed him. The rest of the plasmid completely tanked and failed, and actually you couldn't recover the plasmid from those colonies because it was so unstable. Now, this is where I went and learned. And I learned that a lot of the things I was doing to design this, and this is synthetic biology developing as it goes. We talk about computerized design of synthetic biology, and I encountered this block where I found that actually the computerized design tools that were and are, by and large, still used by people that aren't tied to a license. We'll say DNA 2.0's thing is fantastic for designing DNA, but you must then order your DNA from them. So it's not free form. It's not something you can just use without fear of a blowback. And all the tools that people actually seem to rely on and recommend are now since discredited. The algorithms they use are discredited. The methods that they use are discredited. And that won't happen unless there's the ability for people to do this at low cost. Now, we all know synthetic biology is great, right? It can turn lead into gold. And this was in the exhibit up in Science Gallery recently. It was an apparatus that over the course of Science Gallery's Grow Your Own exhibit actually turned auric chloride into small gold nuggets to kind of make people think, okay, we talk about synthetic biology as if it's alchemy. It actually is alchemy. But the point of this was also to say that while you can make gold nuggets out of auric chloride, it's not cost effective. So you can do alchemy, you can do what the alchemists believe. You can turn base matter, which is found in seawater, auric chloride, into the finest of metals. But no one would actually do that for profit. And it's kind of, I love this message. It was my favorite piece. And it was kind of, a lot of people just walk past it, even though it looks kind of cool. But to me, this is a big problem in synthetic biology, that we're talking about big things, amazing things. And sometimes those things are big, amazing, and exciting, but they don't have a market, and they'll never have a market. And, they mightn't even have a use case. That they're great ideas, but that they're not good in practice. But then you have things which are more exciting and are receiving no attention because right now there's no way of monetizing them. So these are Schwanella nanowires. We talk about biocomputation. These are nanotechnology scale wires connecting cells to one another, and they actually shuttle electricity at fairly high efficiency and low resistance. And the reason the bacteria do this is so that they can dump electrons from their respiration left over. There's no oxygen where they live, so they can't dump their electrons like we do on oxygen, burning it. They dump it on small nuggets of metal that actually grow as they do this because of the reduction. And that's why there's no auric chloride left in the oceans to make cheap gold. Bacteria like these guys made all the gold mines using nanowires to burn energy and dump their electrons. That's where gold comes from. And this is amazing stuff, but at this moment in time, we don't know how to commercialize nanowires. We know they're amazing. We know that someday this will be the foundation of incredible new technology. But right now, it's more or less a curiosity. And there was this, I'm sure Bill will recognize these people because there was an event, the Idea Translation Lab, which is hosted in Science Gallery, where a lot of students got together to kind of do a micro iGEM, where they're supposed to come up with good ideas for a design, engineering, and commercialization strategy for a hypothetical synthetic biology product. And the winners were... there was kind of a split vote at the end. But one of the winning teams didn't have a product. They were suggesting a synthetic biology consulting service. And there is very little in the way of synthetic biology consulting, market consulting, or analysis. And often you cannot know at present. You will not know what the market is like for your idea in the lab until you test the market, which is a whole other layer of blindness as a researcher and a developer. And we loved the idea. And they did everything right. They even had the show. They all had expressions. They wore suits. They were sharp. The guy in the background there even had the nice sleazy marketing look while they were up on stage. Off stage, he was just like totally down to earth, but they had it all down. They were slick. They were your marketers. And it was just such an impact that, again, like the auric chloride thing, this is something... nobody's asked this. We're all talking about applications. None of us are talking about assessment. It's really good to see them there. Now, here's an example of where this is a real-world problem for even big industries. So Amyris have come up in 3 separate talks, including my own. Maybe another missed. Amyris are kind of the poster children for synthetic biology-derived bioproducts, I suppose. But they're also poster children for what I'm going to say here is, like my take-home message in synthetic biology is be agile. You have to be agile because there's such a high error rate. There's such blindness. And the only way to deal with this right now, until there's a better ability to predict, until we've got better protein engineering, better ability to generate empirical data to test your idea without actually having to generate the DNA and test the market, you have to be flexible and you have to be fast. Now, Amyris, of course, artemisinin is going to be a big hit. And I'm glad it's happening, even if I have my reservations about exactly how they're going about, putting all the wormwood farmers out of business. But it is more important that artemisinin's price is now going to crash. And hundreds of thousands of people will probably be alive to see the next wave of synthetic biology because of artemisinin. So go Amyris. But then they got into biofuels. And this is a bugbear of mine because I can't imagine why someone would convince themselves that biofuels can replace petrol. Now, of course, microscale biofuels, as Bill mentioned, fantastic idea. But if you talk about trying to replace an industry that is paid by governments to go to a patch of land and dig up free fuel and sell that fuel at an enormous margin. And you want to replace that with something where you will have to allocate like hundreds of thousands of acres of land to grow feedstock to then make a fuel. I mean, you've already lost. It's not happening. And Amyris got into that game. Now, I may balk, but where I can admire them was when it became apparent that that wasn't working, they pivoted immediately. They said, okay, but we've realized biofuels are not working. They're not going to work for Amyris. They're not going to bring us any significant revenue. We're disappointing our shareholders, etc. Many companies would double down on this, and they'd get behind their idea and say, no, we're going to make this happen. Amyris kind of took a second look and said, okay, we went down the wrong path, let's pivot. And now they're into flavors and fragrances market, which, frivolous as it seems, is, of course, a very profitable endeavor. And as I like to point out, some fragrances and flavors can also have a significant ecological and social impact. Take ambergris. People still hunt and kill sperm whales to get ambergris. If you can synthesize ambergris, you will actually save the whales, which is the pinnacle of human achievement. So I actually, don't write this off just because it's kind of frivolous. Amyris are a great test case in saying, fail fast, fail often, move on. Doubling down is not the way to go. Now, another significant problem to this that, I think we had all sort of looked at with excitement was the first part of that yellow curve is nice and steep. And we were all looking a few years ago going, synthetic biology is about to explode because any minute now that's going to go less than 10 cents a base pair and then less than a cent a base pair. But actually it leveled out. It just went shoo. Because they hit a certain limit in the technology used to write DNA where you could only write a certain length. And the chemical synthesis methods are basically topping out at 200 base pairs with the best available methods. After that, it's all human effort. And this is why the cheapest DNA available is the smallest lengths of DNA, because they don't have to do any effort. They won't do any assembly for you. The less human effort involved, the cheaper it is. So you can get cheaper than that if you go for less than 500 base pairs. But one of the problems is that it remains expensive to prototype, and therefore it remains expensive to fail. And if what you have to do is be agile and be prepared to fail fast and fail often, and I do believe that's the correct path, there's no correct answer here, but the problem is that in synthetic biology right now, the cost of failure is so much higher than in software, obviously, but even in hardware, where you have to order new PCBs or new whatever. Firstly, it's more predictable, and secondly, it is cheaper to prototype. The advantage of synthetic biology, which never comes into the charts, is that if you get it right, you don't need to scale up manufacturing. And that's remarkable, it's amazing, it could be transformative. You come up with one cell that does it right, and you can have as many cells as you need. So the prototyping cost can crucify you in synthetic biology. But if you can get it right, the production costs are so low for a living bioproduct or for a DNA product or a protein product that you can more than make up for it. That's what you're aiming for, the break. Now, I mean, there are companies, among them Cambrian, who claim that they have private methods that can crash this thing, and they draw that chart, and then they just have a nice steep drop. And unfortunately, this has failed to materialize. I mean, even if they kept the secrets to themselves of how they actually do it, I would object. But I wouldn't object enough to turn down 1 cent per base pair DNA. But most of these foundries haven't revealed how they're planning to do this, and they're kind of keeping it very close. So it's unclear. But I do hear encouraging things. I do hear people saying that many of these foundries, like Gen 9, may soon release to the general public services to synthesize cheaper DNA. So it's not all bad. It could actually be that in the next few years, you will get your 1 cent per base pair DNA. And I'm promised that people behind the scenes have visited these companies, come out and seen amazing things. So let's hope that they're onto something. Now, this leaves you with kind of looking at what things are on the market or what things are coming to market, and how did they translate. Glowfish, you can buy these as pets in the US, and they're just fluorescent fish. They come in a variety of colours. And that's it, So where did they come from? They weren't actually explicitly designed as pets. They were part of a research project that was going to be trying to assess water purity, I believe, coastal water purity. So they were going to hook up some sort of biosensor to these guys that they would start or stop fluorescing based on the purity of the water. These guys don't have any biosensor in them. I think they were part of the path towards that product. And somebody came into the lab and was like, man, I'd love one of those. So they commercialize them. And there are other commercial products like that. Actually, there is another pet. You can get an axolotl, which has green fluorescent protein now as well, it came from a developmental and embryonics lab who were just working with axolotls, and someone was like, how much? How much? I'd like a fluorescent axolotl. I discovered this by accident on a pet forum, that they were selling these. It's not even marketed very well. Anyway, so there are actually a lot of products just happen to find their way out of the lab because somebody chooses to commercialize something that was never designed for that. My bugbear is that most plasmids designed for teaching were actually just spun out. So if you buy a plasmid for your first, fluorescent cell experiment, which is what I'm trying to revamp, most of them are actually just cloning plasmids with GFP and ampicillin on them, and they're just repurposed for that. There's nothing designed about them for their case. They're just taken out of the lab, put in a box, and sold. That bugs me because it's not that, proportionally, the amount of effort needed to make a better product. You could take out the ampicillin from the kit if you design the right plasmid. That's what I'm trying to do. But that's like the glowfish. It's just translated because of opportunism. And then you have things that were never designed exactly for a monetization. They just want to get them out. Where's the barrier? What happened? So this is golden rice. It is only part of the solution, but this could help save lives around the world and save eyes around the world. It's an amazing thing. It should be in most of Asia and South America by now. It should be preventing blindness, but it's not. Most of that was because of a patent thicket, which came down to okay, how are you planning to commercialize this and make a profit on it so that we can have our cut as patent holders? And that's where the dark side of translation. And in the end, they came up with a kind of an unsatisfying licensing thing, which says that as long as you're only growing it for subsistence, you can do whatever you like. But if you earn too much, they want their cut. This ugly fella, horseshoe crab, I threw it in there more for the shock value than anything. These guys save your lives all the time and you don't know it. And there was a lovely article again on these guys recently, I can't remember the publication, and they were talking about how they're threatened by the industry that extracts their blood. Their blood is used to test for bacterial contamination in pharmaceutical products before putting them on market. Their blood is remarkably good at giving an instant binary answer. Are there bacteria in this sample? Or is there even lipotoxin, toxins from bacteria in this sample? Amazing property. And these and other animals, like the sperm whales and their ambergris, are under threat because we need their bioproduced products. And this is where you can do social good, like with golden rice, while making a profit. Their blood is worth so much money. If you could find a way to synthesize that, and people are now, you could make an enormous amount of money and save the horseshoe crab. Make enormous money on ambergris and save the sperm whale. So there are areas you can choose. And then there's the stuff that's not possible. And this is when talking about engineering of biology. You can either look at things where you undercut the market for fragrances and flavors. And you can produce far more cheaply, so you can make a better margin and you can put all the other guys out of business. But that stuff's already possible. You're just undercutting them. What about the stuff that's not possible? So spider silk. I put these slides in the wrong order. What if you could have clothes made of spider silk? What if you could have surgical implants coated in spider silk? What if you could have tether lines made out of spider silk? It's an amazing material. A shirt made of spider silk would be bulletproof. So it would be pulled into the wound with the bullet. But, the fabric itself is actually bulletproof. It's astoundingly tough. And it has so many material applications that, if you could produce this, it would be great. But you can't really milk spiders. Someone did at an extraordinary cost to make this, just for the sake of showing what a spider silk thing would look like. But you can't actually economically do that. But they've engineered silkworms that produce, instead of their own silk, spider silk. And on tests, their silk actually compares very favorably with golden orb weaver spider silk. And they're preparing to bring these to market. It's genius because there is already in existence a production process to deal with silkworm cocoons and turn them into silk products. So this is boggling. This is how you do it. Any other way of doing this seems silly. You have the spider goats. We can spin silk from spider goat milk. That's very odd. And there isn't a production process optimized for spider goats. You haven't even, probably most people here, well, maybe this crowd has heard of spider goats, but that's the point. Why have so few people heard of spider goats? Because while they're academically quite interesting, silkworms were where they should have looked in the first place. And this is stuff that was never possible before, but everyone wanted it. And with synthetic biology, you can make it happen. Now, when it comes to funding and making that happen, I mean, I've been lucky, very lucky, and I'm very grateful for that. But how do you get the money to do the expensive part, the prototyping? And the Glowing Plant Project, as much as I have my hopes and my doubts and my misgivings about a few of the things they've done around the way, they raised over $480,000 on Kickstarter. Kickstarter promptly made biotech against terms of service because they got lots of people giving out. But these guys raised a record sum of money to make a plant that glowed in the dark. And that's all it does. They don't even pretend it can actually light the room. But it's inspiring. It's amazing. You know, it's really cool. I mean, the joke is that what they're planning to do isn't what is in the picture. The picture is firefly luciferase with external energy added in a petri dish. What they're talking about is somewhere down the page in a nice humble picture, which is this very delicate blue thing that sort of, if your eyes adjust to the dark, you can make it out. But like, you know, people are mad for this. You know, it's a glowing plant. That's crazy stuff. So 8,433 backers. I should be so lucky. You know, I mean, so there is excitement if you can deliver the right message, which I'm quite bad at doing, as it turns out. There is excitement in synthetic biology outside of labs, and we underestimate that. We kind of talk ourselves into thinking, how will we get people to accept synthetic biology? Rub the beard or, you know, scratch the neck. But then these guys just put out, you know, they got a few people to tweet excitedly. They put a hype title on it, and they had this picture of a glowing plant, and they said, hey, do you want one? And people just got behind it. They vastly overfunded their project. So, you know, this is exciting. I'm not going to say that crowdfunding is always the way to make your project happen, but, you know, there you go. So I don't actually have a great deal of experience personally in translation, but I've made plenty of mistakes. I've observed a few other people's mistakes, and I'm kind of excited to see where the stuff that is currently a bottleneck or a roadblock could soon evaporate, to see what models can help people to make these things real? What can help make it as easy as it was, relatively speaking, to make the Silicon Revolution happen in the 80s? And I suppose I'm optimistic. I hope everyone in this room is optimistic. I hope that this happens in Cork. And I welcome suggestions for how I can raise that much funding on my own project next time. But in the meantime, you know, good luck everyone bringing your own products to market.